Good afternoon, everyone. My name is May, and this is Anna, and we're researchers in the Van Vliet group. We're really delighted to be here today to share with you this image, both for our attendees in person as well as those on Zoom, actually including our families who are zooming in from Poland and Hong Kong. Now, what you see in this image, colored in magenta, are human astrocytes, and these are a key supporting cell type in the brain. These astrocytes are extending their many cellular arms, snaking their way around a 3D printed material shown in red that we developed in our lab. Now, to tell you the truth, like most things in research, we actually discovered this quite beautiful cell morphology entirely by accident, because astrocytes aren't even the main cell type we study in our group. But to understand how we got here, I'd like to take you back to the beginning. These astrocytes represent a profound paradigm shift in biology. You see, these cells, even though they're found in the brain, were actually obtained from stem cells derived from human skin tissue. Our ability to derive neural cells in this way is really transforming our fight against neurological diseases. Currently, there are no cures for any neurodegenerative disease, such as Alzheimer's disease or multiple sclerosis. One of the major challenges this field has faced has been the lack of access to human neural cells with which to study disease mechanisms or to screen for drugs. Although animal models have been helpful, oftentimes those findings haven't translated to human patients. In our brains, we have a rich variety of different cell types, including the main functional unit, neurons, and a rich cast of supporting cells called glial cells, including astrocytes, our favorite oligodendrocytes, and microglia. These cell types all interact in richly complex ways, and those interactions have been implicated in various forms of disease. Now, over the past decade, scientists have gained an unprecedented ability to obtain and derive these neural cell types from human stem cells. And in our lab and many others, we use a type of stem cell called human-induced pluripotent stem cells, or hiPSCs. These are stem cells typically derived from reprogrammed skin tissue, and they're so named because they can differentiate into a plurality of different specialized cell types. For example, iPSCs can become epithelial cells, or blood cells, or neural cells. And in our lab, we can apply chemical factors to coax these iPSCs towards becoming neural cells. Now that we've attained patient-derived human neural cells like astrocytes and neurons and oligodendrocytes, we can use those cells to probe disease processes. And I'll now hand over to Anna, who will tell you a bit about how we use those cells to study one such disease, multiple sclerosis.
A hallmark of multiple sclerosis is damage of myelin, which you can see here in green color. Myelin is this amazing protective and insulating layer that is wrapped by cells called oligodendrocytes around axons, those long arms of neurons that conduct neuronal signals between the brain and the rest of the body. Damage of myelin interrupts the signal, affecting our vision, mobility, and other vital functions. If we could stimulate oligodendrocytes to repair myelin in the process called remyelination, we could reverse the disease. Unfortunately, today we have no cures for MS and no therapeutics to stimulate this process. At the Van Vliet group at MIT, we've been working toward understanding how the microenvironment in MS drives the disease. Toward this goal, we developed a unique platform called artificial axons. In this platform, we use advanced 3D printing and biomaterials to create mimics of axons. You can see those as those red pillars here in this top and side view. These artificial axons mimic the geometry and mechanical properties of real biological axons, such as those that you can see here in this SEM rendered image. This platform enables us to directly visualize and quantify myelin wrapping by human oligodendrocytes. You can see myelin wrapped around these artificial axons in green. We can also watch myelination live. We can now use this platform to study myelination in various conditions that represent the disease, including inflammatory factors, and also in the presence of other glial cells that Ming introduced to you that can also affect myelination. Astrocyte, a star of our awarded image, is one such cell that can affect myelination. Although we haven't studied astrocytes in the past, we now include it as a part of our system and key player in myelin damage and repair. In this image here, you see an example of human oligodendrocyte wrapping a green myelin around artificial axons. in the presence and close interaction with an astrocyte. Our goal is to use artificial axons to discover new therapeutics for myelin stimulation, the repair of myelin in MS. And with this, I would like to thank you and acknowledge our collaborators at MIT and the New York Stem Cell Foundation and funding, and especially thank Erica and her team at Koch Institute for working with us on this image. Thank you very much.